Good evening. Thank you so much for coming this evening to Science at the Theater, sponsored by Lawrence Berkeley National Laboratories Friends of Science. First, I'd like to recognize all those people who are Friends of Science, an organization that's been in the laboratory for a while, and we appreciate you being here and appreciate your continued support. My name is Don Medley, and I am the acting head of public affairs at Lawrence Berkeley National Laboratory, and it is certainly an honor to be here tonight to introduce a stellar panel that we have to talk about the state of research in breast cancer. But first, I'd like to thank a couple of sponsors for tonight's event, sponsors who've been with us throughout the entire Science of the Theater program: the University of California, Berkeley, Chabot Space and Science Center, the Exploratorium, Osher Lifelong Learning Institute at Berkeley, Lawrence Hall of Science, and the science departments at Albany, Berkeley, and Oakland high schools. And also, for those of you who are new or have never heard about Lawrence Berkeley National Laboratory, we're the lab on the hill above campus. We are the oldest of the national laboratories in the United States. We have a very distinguished scientific pedigree with 11 Nobel laureates. Our scientists make up 4% of the National Academy of Sciences, and we have a very close and long relationship with the University of California, Berkeley, of which we're very proud. We are very fortunate tonight to have an extremely dynamic panel of Berkeley Lab scientists who will discuss research advances related to susceptibility, early detection, prevention, and therapy of breast cancer. They will describe a biological systems approach to tackling this disease from the molecular and cellular levels to tissues and organs, and ultimately to the whole individual. We also are very excited tonight to have a very distinguished moderator who I'll introduce at the end of the list. Our panelists include Dr. Joe Gray, the Berkeley Lab Life Sciences Division Director, who has pursued several aspects of cancer throughout his 35-year research career. Joe received his undergraduate degree from the Colorado School of Mines and a PhD in nuclear physics from Kansas State University. It was during his time in the Biomedical Sciences Division of the Lawrence Livermore National Laboratory that he became increasingly active in cancer research. He has worked at UCSF since 1991 and became the Division Director for Life Sciences at Berkeley Lab in April 2003. Dr. Gray's current research program involves analysis of the genetic progression of human breast and ovarian cancer. Tonight, he will share his research on how chromosomal abnormalities contribute to cancer and respond to gene-targeted therapies. Our next panelist is the distinguished Mina Bissell, who is a distinguished scientist at Berkeley Lab and is the former Life Sciences Division Director as well. Simply put, Mina is a pioneer in breast cancer research and has played one of the key roles in changing long-established paradigms about cancer and the reasons they grow. She earned an A.B. with honors in chemistry from Harvard Radcliffe College and a PhD in bacterial genetics from Harvard University. She joined the Berkeley Lab in 1972, became Director of Cell and Microbiology, Molecular Biology in 1988, and was appointed the Director of Life Sciences in 1992. She is also the Office of Biological and Environmental Research at DOE's Distinguished Scientist Fellow in Life Sciences. Tonight we will hear about her approach to the challenge of breast cancer from the breast's three-dimensional tissue microenvironment and how the intracellular conversation triggers malignancies. Our third panelist is Mary Helen Barcellos-Hoff. She is the Deputy Director for the Life Sciences Division at Berkeley Lab, and her research identifies what exposure to ionizing radiation can tell us about how normal tissues suppress carcinogens. She received an undergraduate degree in biopsychology from the University of Chicago in 1978 and a doctoral degree in experimental pathology from the University of California, San Francisco. Her postgraduate training was conducted at Berkeley Lab with Mina Bissell and concerned the impact of extracellular matrix signaling on mammary epithelial functional organization and differentiation. Today, the overarching goal of Mary Helen's research is to understand how radiation affects multicellular processes during neoplastic progression. This will lead to a better understanding of the risk of radiation exposure, the mechanisms underlying cancer development, and will certainly have some surprising implications for radiotherapy. And all that was a mouthful. To moderate our panel tonight, and from previous meetings, I don't offend anyone, but the panel does need moderating. Dr. Susan Love, a breast cancer research pioneer, author, and entrepreneur, is the president and medical director of the Dr. Susan Love Research Foundation, and founder and medical director of Windy Hill Medical, a breast cancer prevention company. She is a clinical professor of surgery at the David Geffen School of Medicine at UCLA. Dr. Love received her medical degree from SUNY Downstate Medical Center in New York, did her surgical training at Boston's Beth Israel Hospital. She has retired from the active practice of surgery to dedicate herself to the urgent work of breast cancer prevention. Her one goal for the Dr. Susan Love Research Foundation is to eradicate breast cancer within our lifetime. She strongly feels that we now have the tools we need to get to where breast cancer starts and only need the will and resources to make prevention a reality. Again, welcome to this evening's roundtable discussion, and now I'd like to turn it over to Dr. Love.
Over here. Thank you very much. This is really, I'm very excited to be here because to me, the most important thing that's driving all the changes in breast cancer is the science. And tonight, we have the advantage of having some of the people that are at the forefront of things I talk about all the time. So I'm as excited to hear what they have to say as you are. My job, I think, to a certain degree, is to both moderate and translate. So I'm going to try to set the stage a little bit before each talk about how this relates to what we see in breast cancer and in the clinic, and then help ask some questions afterwards. And then at the end of all the questions, then we'll be open for questions from the audience. You might want to write them down. At least if you're like me, you'll forget by the end of the talk what your question was at the beginning. And we'll have time for everybody. There have been a lot of changes in the way we think about breast cancer, clinically and certainly scientifically. But I don't think that they've all really reached the public arena. And a lot of times what you're reading about in the press and in books are sort of the old theory, and there's a new paradigm in which we're thinking about it, and that's what you're going to hear about tonight. So one of the first things that's really changed a lot is we've started to realize that this idea we had about cancer was that cancer is one kind of cancer and slowly grows and starts as a grain of sand and then becomes a BB and then becomes a lima bean and then becomes a walnut and then a lemon and then a grapefruit and then a melon. If you could just find it early, you could cure it and everything would be great. So this whole early detection notion was based on the fact that cancer grew at a nice even rate and that all cancers were the same. And in fact, our treatments for breast cancer until very recently have pretty much been one size fits all. We don't really care if you're how you are or how the cancer is. Everybody gets the same combination of surgery, radiation, and whatever the chemotherapy is of the day. And sometimes it works, and sometimes it doesn't. What's really changed is we finally realized that all cancers are not the same. So instead of just looking at them under the microscope and saying there's a lobular and there's a ductal, there are these two kinds based on their appearance, we're finding out that if you look at the DNA in the cancers in these things they call microarrays, where they can look at 1,000 different mutations in 1,000 different cancers at once, and they put them all, it's actually a little tiny thing, but I have to think about it as a big thing. They put them all in these little wells and then put it, It turns red if the gene is turned on, or green and red if it's turned off, and they go blah, blah, blah. And then they have all these different cancers, and they find out that there's 6 different kinds of breast cancer, at least 6 that we know of now, based on their DNA patterns. So there's one kind that's a good kind, that they call it sometimes the almost normal kind. Probably doesn't matter when you find it, you'll do fine. There's one kind that's really a bad kind. Probably doesn't matter too much when you find that one. That one's not going to do very well. And there's different ones in the middle. What Joe Gray is going to talk to us today is the science behind this. Gee, there's a lot of different kinds of breast cancer. And the implications of that are not only that the prognosis might be different depending on which kind you have, but that the treatment might be different depending on which kind you have. So if you match the treatment directly to the kind of cancer you have, you'll have a much better result than if you give the same treatment to everybody and only some people are going to respond. So this is really, this type of science, this science that he's going to talk about is really what's driving now what we're seeing in the clinic in terms of much better results in terms of cancer treatment, because we're doing much better at figuring out who should get what based on what kind of cancer they have. And with that, I will hand it over to Joe Gray.
Well, thank you, Susan. I have to remember the next time that I'm asking somebody for money and they ask what we do with all of this microarray data. I've got to get that.
Yeah.
I missed that. That's much better than the way I say it. Okay. So I am going to start with the theme that Susan introduced, and that is the idea that not all breast cancers are the same. And we've learned this over the years, basically from a very large number of clinical trials where it's become apparent that this one-size-fits-all strategy doesn't work. In fact, the vast majority of cancers will not respond to many of the agents that we bring to bear, but some small fraction will actually do quite well. And so one of the things that we try to do in my business is to understand what the molecular basis is for that kind of differential behavior. So what we're going to try to do here in our research program is to first try to understand who won't respond well to therapies and who will. The reason that we want to find out who does not going to respond is because many of these treatments are toxic, and so we would like to be able to change rapidly, maybe even before we start, so that we can identify some that will be effective. Having identified those patients that are not going to do well, what we want to try to do is to identify or develop new therapeutic approaches that will be effective in these patients. And then eventually, as we get more and more of these effective therapies, what we would like to do is to develop assays, molecular assays, that will allow us to, in fact, individualize these treatments to the various patients. So I'm going to start and take you through just a few vignettes for each of these areas. And the first one of them is to make the point that, to first approximation, cancer is a disease of the genome, of the genes, of the DNA that make up the cells in the breast and other tissues of the body. Now, I'm going to focus on the genome of the cancer cells themselves. When Drs. Bissell and Barcellos-Hoff stand up here, they're going to give you a slightly revised version of what it is that I'm going to tell you, based on the concept that it's not just the cancer by itself that's important, but I'll focus on the cancer. And so if you think about a cell that might become a cancer, what you appreciate the fact is that the coding material, the thing that tells the cell how to behave, is encoded in the DNA. DNA gets transcribed into this a molecule called RNA, and that eventually gets converted into proteins, which make up the components of the cells that we had to deal with. And a cancer arises typically because of damage somewhere in the DNA. Now, so what have we got to work with here? One of the things that's remarkable is that every cell in your body carries 2 copies of 3 billion base pairs of DNA. That is the genetic code that tells the cell how to behave. And cancers arise because this DNA gets corrupted in a variety of different ways. The DNA sequence itself can be changed. You can chop up the DNA and make too many copies or too few copies of different genes. You can change the way the DNA is read out. And all of these things can contribute to the carcinogenic process and also to the way that the cancers behave. Now, we've known this for 50 years. What we haven't had is the ability to actually scan this entire set of DNA and find out what's going wrong. And one of the things that Susan mentioned is that in the last really 5 years, we've gotten to the point where we are now able to essentially scan the entire 3 billion base pairs to look for what's going wrong. And the technologies that we use to do this are really born out of technologies that were developed in the semiconductor business or that take heavy advantage of new emerging nanotechnologies. But the bottom line is that we can look at all of the genes. Just as an example, some of the work that we do in my laboratory, this is a plot of the number of copies of a particular part of the genome as a function of distance along the genome. So you
It's
can
sort
think
of
this
blah, 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 like that. It's sort of like the blah, 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 blah.
Yeah, this is the blah, blah, blah part of it, yes. How you get that is that. But what, if this were a normal tissue, this is a breast cancer. If this were a normal tissue, you'd see a nice flat line along here. But what you see instead are all of these squiggles going up and down. And when you see a squiggle going up, that means that there's too many copies of that gene. And when you see a squiggle going down, there's too few of that copy of the gene. And interestingly enough, for those of you who pay attention to the breast cancer field, one of the genes that's particularly important in breast cancer is this gene called ERB2. What you can see here is this cancer has too many copies of it. So this gives us a very quick view of the entire genome, and we can do this at a variety of other levels as well. But it tells us what's gone wrong in this cancer. And back to the point that Susan made before, we've measured hundreds of these, and we've never seen two that were the same. There are many common features, but in detail, they're all different. So this is one of the reasons why dealing with breast cancer and other cancers is actually quite complicated. Well, what do these things do? What do these abnormalities do? There's going to be a test at the end. Everybody will be required to regurgitate this. So what we've learned from 50 years of biology is that these changes are influencing the way the cells regulate themselves. And we have quite a wiring diagram that we're beginning to understand how this happens. A lot of what goes on involves communication between the interior of the cell and the exterior of the cell. Dr. Bissell and Barcellos-Hoff will come back to that. But we now know some of the genes in this process that become corrupted. And one of these is this ERB2 that I was just telling you about. That's located,
which is also called HER2-neu.
Called HER2-neu.
Because clinically, it's more commonly called HER2 or HER2-neu, just for the people who might have heard of it.
Good. All right. So anyway, when you get too many copies of this, you signal too intensively to the interior. That helps to potentiate cell growth. And one of the things that this kind of knowledge allows us to do is to develop candidate therapeutics against these cancers. And so one of the ones that is now clinically available is a drug called lapatinib. There's another one called Herceptin, but the bottom line is these kinds of curves are survival curves. This is really the fraction of the patients that are surviving as a function of weeks or years after treatment. And the higher the curve is up here, the better patients are doing. And what this shows you is that patients that are treated with this drug called lapatinib, which attacks this gene, actually do better than if you don't have that treatment. So it's taking us in the direction of understanding how it is that we can make better drugs, and you can actually develop assays that tell you which patients to treat. So each of these little green spots indicates a cell that carries one copy of this gene, and a normal cell would have two, and you can see here that there are hundreds of spots on it. So this just tells you that this patient ought to be susceptible to this drug. Well, this is a big business. We're very excited about the idea that we can understand what goes wrong and develop drugs to treat it. What we have underway right now is an amazing business out in the pharmaceutical industry of developing these kinds of targeted drugs. And in fact, there are over 400 drugs that are now currently being developed against some of these targets. The problem is, how do you know which ones to apply to which patient? And because we're still early in our understanding of this, one of the things that we've discovered is that this is a very expensive process. It may take as much as $1 billion to get one of these drugs into the clinic, and about 80% of the time, or maybe even 90% of the time, the process fails. So we need to have a better way of understanding how to do that, and that's what some of the research at LBL is trying to address. How do we identify who's going to do this best? And so we've been spending a lot of time developing experimental cellular models that we can manage in the laboratory. And we use these models of individual breast cancers to try to test the efficacy of this large collection of drugs. Now, this is a lot of experimental work, and so one of the things that we've invested in is the development of automation through the LBL engineering department that helps us do this efficiently. And our goal is actually to be able to do this for every drug that's out there and available, and we're beginning to do this. Why is it hard to actually understand who's going to respond and who isn't? Because of this heterogeneity between tumors. I showed you these kinds of little plots before, and one of the things that I've done here is to choose 3 breast cancers, all of them amplified for this ERB2 or HER2 locus. But if you look at the rest of the genome, you can see that they're all different. And so this is one of the reasons why even if we have a drug that's targeted to ERB2, it won't necessarily work all the time. And so we've been busily analyzing these model systems, asking, can we learn even to refine our treatments within this population? And I won't go through this complicated slide in detail, but the bottom line is that we've now been able to take these models that we've developed in the laboratory, identify some molecular markers that we think would predict whether or not a patient would respond to this drug, lapatinib, that I was telling you about earlier, and we've applied them to the patients that are ERB2 positive and therefore we think should respond. The ones that we predicted to be sensitive do respond. The ones that we predicted to be resistant don't respond. So it's the beginning of a strategy that we think will lead us to the point where for every drug we'll have a set of markers like this that will allow you to say you will respond or you won't respond. And so that's the gold at the end of the rainbow here, and I think that we're making good progress in that direction. So basically, to summarize what I've told you, some of the modern analysis tools that we're developing really do allow cancer genomes to be scanned comprehensively for the aberrations that drive the cancer. We've got hundreds of new therapeutics under development that we can use to attack these abnormalities, and we think we have the analysis techniques and the models that will allow us to make molecular assays that we can use to guide these drugs effectively into the individual patient. Thank you.
So I just want to ask you a couple of questions before I let you off the hook. All those pathways, if you just block one pathway, aren't there alternative routes? I mean, isn't it like when the freeway gets clogged, you can go on surface roads?
That's exactly the problem. There are multiple pathways. You could actually see them in the diagram that we had up there. And in fact, one of the interesting things about that last little example that I showed you, I mentioned that we were able to predict some patients who would not respond. But what we learned from that analysis was that the reason that they won't respond is because some of those other pathways are on. And so it's just a bypass pathway. And so as we go forward in treatment, what we're going to have to do is to not treat with one agent, but to be able to intelligently combine them in ways that start to block the whole thing, the whole network.
Yeah. And then my other question is, well, what about, can they become resistant? So you treat them for a while. Certainly we find this with antibiotics, for example, and then you get new bugs that are figured out how to get around it. So will that happen?
So absolutely, that will happen. One of the things that's remarkable about cancers is you get all of these abnormalities because the cancer cells, amongst other things, have lost the ability to control the way their genomes get parsed out during cell division, so they're very unstable. And so this gives us a strong mechanism by which they can evolve resistance. And so again, the more drugs that you can treat with, the higher the probability there will be that you can counter that resistance before it develops.
Okay. HER2-neu is about 20, the people who overexpress ERB2 or HER2-neu, why it's a long story why it has two names, but are only about 20% of all breast cancer. So while it's really exciting that we have a nice target for them, it still leaves us with 20%. There's another group that are sensitive to hormones, and there we're able to use things like tamoxifen and the aromatase inhibitors to treat them. But interestingly, not just right along with what Joe is saying, not everybody who's hormone positive actually responds to hormones. Some of them, even though they look like they have the right marker, don't. And so parsing this gets to be very complicated. And my worry is we're going to have to put so many multiple drugs together to answer the problem that maybe we should prevent it first.
I knew you were going to say that.
Thank you, Joe. So next is another new paradigm shift. So one paradigm shift we've got is that there's more than one kind of breast cancer. Another paradigm shift, I will explain that in a second, or not, either way. You can show them or not, whatever. But the other thing is, we used to think about cancer as if the cell was almost, and this is all cancers, not even just breast, came from somewhere else. Like it invaded from another planet or something and took over our body. And then we had to somehow fight it off. And you think about all the words about cancer, they're war words. We have to battle against cancer. You look at the obituaries, they lost their battle and the war against cancer. But you know what? These cells are not foreign invaders. They're our own cells that go bad. They're not Osama bin Laden, they're Timothy McVeigh. They're our own terrorists. We grew them ourselves. We grew this terrorist all by ourselves. We didn't come from somewhere else. And so part one is, as opposed to bacterial infections that are coming from somewhere else where you need homeland security, these are our own cells. And the second part of it is that these cells are not in isolation. So in the old days, the way we studied these cells was we looked at we took the cells and we put them on a petri dish and we watched their behavior. And it was a little bit like taking one of you and putting you in an isolation chamber and then studying your personality without letting you talk to anybody. I mean, obviously, we interact, and our personalities depend on how we interact with, who we bounce off of, and all these things. Well, the same thing with these cells. These cells are not in isolation. In the breast, they're sitting there, and they've got fibrous tissue and blood vessels and fat cells, and they're all having a conversation, and they're interacting with each other, and they're influencing each other. And the one who really was the first one to sort of remind people about this is our next speaker, Mina Bissell. And these cells, this is a duct, a breast duct that she has kindly, if I can get this right, it's up there somewhere. Is it this way? Maybe it's down here. No. Anyway, whoops. Nope, that wasn't it. That was there we go. The top red one. Okay, thank you. Okay. High tech. I'm much better with the blah, blah, blah. I'm just a surgeon, but I try to understand the science. So this is a breast duct. Inside you have the ductal cells, and then you have lining cells that hold it together, the myoepithelial cells here, and these are fat cells, these big open ones. But there are also fibrous cells, there are blood vessels, and all breast cancer starts in these lining cells of the milk duct. But they're not in isolation. So that although Joe says that it all starts with abnormal genes that you either inherit from your mother and father or acquire from the carcinogens in our environment, it's not just having the screwed up cells. You need to be in a screwed up environment. And I come from LA. The example I like to use is that is a screwed up environment. But if you took somebody out of South Central LA, you've got a bunch of kids that are really in trouble. And if you take them away from the pushers and away from the gangs, some of them just have personalities and they're going to be in trouble no matter what. And they're the totally screwed up genes that Joe is studying. But some of them, if you take them out of that environment and put them in a new environment, even though they're the same kids with the same DNA, they can behave differently. if they have a different peer group and if they have a different environment. And that's the brilliance that Mina was able to show and will talk to us about, is how we can actually, by changing the environment that these cells are in, these ones that have all these DNA, corrupt DNA, how we can actually change their behavior, and that may be as important as whether they get the mutations in the first place. So just to continue this story, because something that is close to my heart, my research is really on the milk ducts, but the breast starts with this little bit of duct, and we're all, men and women all have this. So when you just have the nipple and nothing else before puberty, we all have just a little bit of ducts. In mice, and this is a picture from mice, they only have one. We have about 6 to 8. And this is the fat pad it lives in. And then all of a sudden the hormones start at puberty. On your mark, get set, go. And when they start, it starts growing. And the duct grows and grows till it fills the whole fat pad. And the duct is this part, and then these are the lobules that make the milk, and the duct is the pipe that carries it to the nipple. And all breast cancer starts in either the duct or the lobule. We think it starts sort of at this junction here, but it may also be able to start in here. And then you get a little more, and then pregnancy. So now the lobules are getting ready to make milk. Lactation, they're pouring the milk out. That's really what the breast is there for in the first place. And then afterwards, it all dies off and gets ready for the next kid. And so that's here. That's the purpose of the breast. But because the breast is the only organ that we're not born with, if you think about it, it's the only organ that's not born fully functioned, that develops later. You have your ovaries, your testicles, all that, you're born with them. But the breast shows up later. So it has to have a lot of cells that divide and stem cells to help it grow and evolve and then get ready for the next kid. And that's, I think, its fatal weakness and why it's so susceptible to cancer. Because it both has all these capabilities, but the more capabilities means it also has more weaknesses. So with that, I'm going to pass this over to Mina, who will talk to us about the neighborhood, not just the kids, but then the environment and the peer group that they live in and how that influences them. Yeah.
Well, you're great, Susan. Very happy to be here, but if I stood behind this, I disappear. So I have to walk. And I'm very happy to be also as part of the science forum here. So I'm going to try and in 10 minutes tell you something that has taken us 30 years to convince the scientific community. And what is that? Is that cells can become a cancer, and yet you could trick them into thinking they're not. Or you can take a normal cell and mess it up, and is going to think it's cancerous. And of course, it's not going to invade the way the cancer cells do, but there are a lot of things that the cells are capable of doing, and I like to say that this is a hopeful view of cancer. Because even if you get it, if we knew how to deal with it, we could do something about it. So how do you become who you are? And how does your nose doesn't turn into your elbow if you get up one morning? Every cell in your body has the same genetic material. And of course, I work at Berkeley Lab with all these physicists and engineers, and I have been there for a long time, and they always wanted to know what the heck I do. And of course, they wanted to know this in 2 minutes. So here is Biology 101 that I give to the physicist. So when your mom and dad met, here you have a fertilized egg. This is the egg, and that little thing is the sperm. So they find each other, they get fertilized. Now the embryo goes and does this, starts growing and growing and growing. And as you know, it becomes an embryo. And before you know it, it does this. So, now, the lipstick notwithstanding, this gorgeous lady came from a single cell. Every cell of those billion cells that Joe talked to you about all have the same genetic material. Not only do they have the same genetic material, we now know from having made a dolly, you all heard about that, right? They took a cell from the mammary gland, they could manipulate it, they could put it back into this thing, and then they get a whole sheep. How did they do that? The cells already were a mammary cell. So that tells you that the cells have the capability of doing something else. So the question I raised for you in the beginning is not an idle question. Every day you look at yourself in a mirror, you are an absolute miracle. Because your nose is remembering to be a nose. Your breast, your prostate, so the guys would listen, breast and prostate have a lot in common. So why is it that we not only arrive at these incredible tissues, but we also keep them the way they are? And I like to submit to you that this problem is very important to cancer, because it's precisely when a cell doesn't know that it's supposed to be a nose or a mammary gland or a prostate cell that it actually starts piling up and goes elsewhere. So I started working, I was a chemist, as you heard, and then a bacterial geneticist, and I hadn't taken any biology, and then I started coming here and working on what is known as a Rous sarcoma virus. This is the first oncogene, or the first cancer gene of a virus that was discovered in 1911. It makes this very ugly tumor on a wing of a chicken. And the guy who discovered it, 50 years later, won the Nobel Prize because he could then make a filter out of this, make a solution out of that tumor, add it to normal cell and show that the normal cells were able to become malignant. And then if you injected those cells back into the chicken, and you again got this. So this didn't make much sense to me because I thought you have billions of cells in your body. If one single mutation could make a tumor, you would be a lump. You wouldn't be who you are. Because we go out in the sun, we get mutation. We eat and we breathe and we do whatever, we get mutations. So when I got my own laboratory, we started doing some really crazy experiments. We took this virus and injected it into a chicken embryo for reasons that I don't have time to tell you. And then we showed that the virus in the embryo was unable to make a tumor. And then people said, oh, that's because the virus is not showing its oncogene. That's not true. We took the oncogene, attached it to this blue thing, which is a marker of the oncogene expression or the cancer gene expression. And you see this very, very pretty feather with all these blue cells. Every one of them could be cancer, but they're not. So the same virus that is making that ugly tumor in the chicken, in the embryo is not doing it. Why is that? Now, if we take this embryo and dissociate it with an enzyme and separate the cells and make them like those single cells Susan was telling you, and we put them in a dish, overnight, they remember they're a cancer cell. So that was very exciting to me. And that and a whole lot of other things told me that indeed context, i.e. the microenvironment that are around these cells, can tell the cell what to do. Because you have the virus in the chicken, you get a tumor, in the embryo you don't, you dissociate it, you put it in a dish, you get mass transformation. So, if the microenvironment does this, how does it do it? So it took me almost 30 years, wonderful postdocs and graduate students who have worked with me to actually show this. And so I began to wonder about that question that I posed with you with Madonna, and that is, how is tissue specificity maintained? How does your nose remember to be a nose? How does your breast remember to be a breast? Why does it turn into your knee or whatever? how does one study this problem in higher organism? Because people had studied these things in single cells, in bacteria, but we wanted to study them in higher organism. How does the program lost in cancer, as I told you, and also in aging? One of the things that changes in aging is the structure of our breast. Is a whole lot of other things that changes, and aging is a very important risk factor for cancer. So, finally, how can we use this information for therapy? So we began to use the mammary gland that Susan so nicely showed you in the beginning. Now here is a human breast, and I'm showing only one of those ducts, and as Susan told you, we have 6 or 7 of them. So the human breast is also like the mouse breast and fills the fat pad, but the human breast itself is quite complicated because it has a duct, it has the nipple right here, it has all these secondary branches, it's like a tree in autumn, it has tertiary branches, and at the end of these things, again as she showed you schematically, is this thing which we call an acinus. And she showed you one of these things where it was in a whole sea of fats which the women's breast has. One layer of epithelial cells, one layer of these other cells, and a layer of something that is called extracellular matrix or basement membrane. These are large, huge molecules, and the things you hear about them is something like collagen. They keep telling you collagen is good for you, and buy yourself a cream, it has collagen. Don't listen to them. You can't add collagen from outside to your face. But the important thing is that collagen and a lot of these other important large molecules that people thought were only scaffolds, in fact have information and in fact are telling these cells what to do. So we then said maybe we want to understand how this particular tissue is formed. So we took the cells from the breast where they are making milk from a mouse. We put them in tissue culture. And when you look at them in the breast, they look like this. They're very pretty. They have a very organized structure, very organized nuclei, very organized secretory activity. You take these cells and you put them in a culture dish and you give them all the hormones they need to make milk and to look like this. And within 3 days, that's what they do. They completely lose it. So 3 days ago in the mouse, they were making all these gobs and gobs of milk and they were looking so pretty. Now they do this. So we argued that maybe that extracellular matrix that was in vivo and wasn't in a tissue culture dish had information. And we showed it with, in fact, Mary Helen Barcellos-Hoff, who is our next speaker, and she did beautiful work in my laboratory. She took these cells and put them in this material that is like a gelatinous basement membrane. It makes the cells think they're like an embryo. They pull this thing over them, they organize themselves, and you see that in about 4 days, they're beginning to look exactly like what was in vivo in the mammary gland. Here is one of those acids in vivo. Here is what we can make in tissue culture. This thing secretes gobs and gobs of milk. But when they were on a dish, they had forgotten to do it. So when we give them the form, the function comes. So we said this is very exciting. We can make these beautiful structure. These are actually nuclei. This is a very lovely cytoskeleton. And this green thing is a basement membrane. So we can make these lovely things in a tissue culture. They make lots of milk. And here the red is milk. The blue are the nuclei of a single cell. This whole thing is a unit and has all this milk. And you can imagine, for somebody who has been in the lab for 35 years, you can imagine how old I am. So I had one of these birthdays, and the kids did this. Yeah. So we have lots of milk. And the question is, how do we do that? How does the milk actually get produced? And does any of this, in fact, is applicable to humans? So I started doing a wonderful collaboration with a guy at Denmark. He's an MD, PhD, and in fact, Susan can do this for me, we can take cells from women who have large breasts. They call them reduction mammoplasty. And we said, could we put these cells in a dish? And could we actually see if the human cells would do what the mouse cells do? So when we did that and we put them in a dish, again, they do this. They look like what 90% of scientists, now maybe 8% are doing what I'm doing, but a few years ago it was 99.9999% were doing this. But as you see, this doesn't look anything like that. So we put them in this gelatinous three-dimensional matrix. The cells organized, they made this beautiful thing, and even the sizes are the same, and you see that it's very similar to human. So we then use this concept to develop an assay. An assay Joe talked about. An assay is a method of determining and measuring something. So we developed an assay. We said, could the normal cells remember that they're supposed to do this? And could the tumor cells forget and make a different form? And then all of a sudden, rather than having to inject them in the mouse, we can actually look at them. So when we did that, on the top, here is a movie. On the top, we start from single cells, and you see every one of those single cells make this beautiful three-dimensional structure. These are not single cells. They are that. Those are the nuclei, and they look like that, and they look very pretty, and they look like what they are in the breast. Down here, every one of these makes these ugly masses. Every one of them look like a tumor. So we all of a sudden have a way of measuring normal and malignant human cells outside human body. Thank God we can no longer do experiments in humans. So you can take these things and you can do all sorts of things with them. You can measure them, you can study them, you can... So we then said, this is funny, because when the cells have the right structure, they appear to know they are normal. When they have this kind of a structure, they're a tumor. And we said, could we manipulate them to make them think they're normal? And we found a way that I don't have time to explain. I'm already going on and on. So we then gave them a molecule that is involved in various kind of a pathway that the normal cells know how to use, and the tumor cells use it, but they're a little out of control. So we reduced the level of that particular molecule in these to that. And we said, what do they do? So if you look at the higher magnification of the non-malignant cell, these are the nuclei, and they have this acinus. And here are the tumor cells, and these tumor cells were actually even derived from those. You see that they're a mess. And we said, could we make these cells remember or forget that they are malignant and make them look like that? And we gave them this molecule, and look at that. So the cells reverted to a normal phenotype. Clearly, they have all those mutations Joe talked about. All those ugly things going back and forth and changing, and look at them. They stopped growing. They clearly are not 100% normal, because you see the shapes are a little different, but the sizes are the same. If you take these things and inject them into mouse, no tumors. Now, if you separate them and add a little bit of this material, the tumor level, it goes way down. And to show you a movie of this, on the top are the tumor cells. On the bottom are every one of those cells treated with this material, and every one of them remembers that they should do the right thing, and they change, and they think they're normal. This was, of course, very exciting. We now have, we then showed, and of course, I don't have time to show you this one. It's a little complicated, so please forgive me, because I'm not going to talk about this. Or maybe I should, I don't know.
No,
should I?
Keep going.
Go
no, keep going.
Should I say?
No, faster.
Okay. No. All right. So when you put these cells in a tissue culture, and you do them with this material, without this material, they look like this ugly tumor. When you give them this material, they organize themselves. And all the molecular biologists and geneticists said, they have to have mutations. Something should have happened. It's sort of, and I said, what? 10 to the 6 of these cells get mutation? No. So we took these cells, dissociated them, put them back in a dish. I have a cold. So without the antibody, they do this. With this particular thing, they do this. And so clearly the genome is a mess, but the cell behaves normally. Even though the genome is the same, they are doing the same thing, but keep remembering the genome here, the genome here, the genome here, the genome here are all the same. But they're clearly different tissues. So tumor cells can do this too, which is very exciting. Okay, so we made a hypothesis and we said we should be able to make the cells normal if we in fact do the right thing. And if we take a mouse, without giving them any oncogene, we can make them make tumors if we destroy the structure. And we have done all of that, and I don't have time to tell you. So, what we have done, what is happening? So form and function are related dynamically and reciprocally, and when you mess up one, you mess up the other. And cancer is both a problem of the genome, as Joe said, and a problem of a structure, as I'm saying. There is a reason your nose behaves like your nose. There is a reason your eyes look like your eyes. There is a reason your liver does the liver. So there is real intelligence in evolution. Okay, we now can take these cells and can revert them in many, many different ways, which is wonderful. And when we do this with one, we actually correct the whole thing. Now, when two higher organism cells come together, the result is not one cell and one making two. The result is logarithmically different. The cells make a junction, they have a bottom, they have a top, they have an organization, all the molecules are in the right place, and so that's why your brain and your body talk to each other. That's why you are a whole. And every time we try and separate part of you, the other part of you doesn't work. And so now people are talking about the fact that we still have not cured total cancer deaths, even though we are doing very well with breast cancer, while, for example, we have done so well with heart disease. So I argue that the reason is that we have been looking under the light. And so I usually show this picture of this very beautiful woman. I don't mean to depress you, but she had BRCA1 mutation. She worked at Berkeley Lab. She actually modeled this dress 2 weeks after she had a breast removed, and she was one of the most feisty, wonderful people. And I like to say that people in my lab work so hard, we all work so hard, because it was too late for Sonia, and I hope it won't be too late for my granddaughter. I have 4 grandchildren, but only this one. So I hope when she grows up, Susan and I and Joe and I and Mary Helen and everybody else would have solved this problem. So where do we go now? As Joe said, cancer is not a single type. There are many different diseases. We have all these drugs to test. How do we do that? So I have been going around fussing with people to test the drug on human cells on 3 dimensions, because that can give us a very good idea of which one of these combination drugs are actually going to do the trick. We are very excited about it. And this assay, as Joe said, here is a good cancer. You see, we put it in this gel, and the good cancer does this, and the bad cancer does that. So we don't even need to do a lot of fancy stuff. We just need to look at them. So with all of that, I would say that these concepts are also important in chemotherapy. The cells behave to chemotherapy depending on the shape of the tumor, and again, I don't have time. So organ specificity or tissue specificity is plastic, dynamic, and context dependent. Homeostasis, the thing that makes you who you are, is all about balance in signaling and is not absolute. And these things are important for tissue structure and not absolute. Thus, half of the secret of the cell is outside the cell, and these concepts are important for understanding cancer. And I like to leave you with this, because I show it at the end of my talk, and I tell it to all these graduate students. I say, you know, thank God for Berkeley Lab, and thank God for Department of Energy. They actually gave me some money. The National Institute of Health thought I was crazy. So I used to be this cat, and this was the establishment, The funny thing is, I now have become the establishment. So I always say to the kids, don't listen to me. Always think outside the box. It's very important. We still don't know very much, and there is so much to learn. And finally, as Joe showed you in the beginning, this is Lawrence Berkeley Lab. This beautiful structure was the first top of a thing where they did the physics. They kept it, and now they do structural biology. And I usually show this to people to not mix us up with Lawrence Livermore. They don't have this beautiful view, and they don't have advanced light source. And I say to them, what does this remind you of? Looks like a breast, right? So you will never forget.
Let me ask you a couple questions.
Thank you.
I have a couple of questions, because it's so exciting that you can reverse the cells, but then we really don't quite have a way to do, well, we probably do that in people, but we don't really know we're doing it. So
Yes.
let me ask you this. The recent data, when they showed that when the hormone replacement therapy, the estrogen plus progesterone, the year that the study came out and everybody stopped, the incidence of breast cancer went down 15%, pretty quickly in 1 year, and only in postmenopausal women and only in estrogen-positive tumors. People said, how could it be so fast? It can't
That
change
explains
mutations. But it must be doing this, don't you think?
Exactly.
Yeah.
Now, I want you to know that those results are because these women were postmenopausal, they were taking estrogen plus progesterone.
I said that.
Estrogen alone, I know you did, but I want them to listen, because estrogen alone is very good for your brain and your breast. And by itself, now Kaiser literature says this, 7 years after you take estrogen alone, it doesn't increase risk of breast cancer. I have taken it for 18 years, so.
We don't totally agree on this, Mina and I. But
yeah, I know.
But it is true
well,
that
we can fight it out after.
Yeah, but it is true that estrogen alone is not the same as estrogen plus progesterone. I totally agree. But that's my point. And
Yeah.
also, if you start on tamoxifen, your mammogram gets less dense. Okay, what causes the density? Well, it's not the cells, it's the neighborhood.
That's exactly right.
neighborhood.
Yeah. It has a lot more collagen, and it has
But the problem is, now when are we going to be able to apply this directly to, instead of being able to say, oh, that must be what's going on, when are we going to be able to consciously say, okay, you have cancer, this is how we're going to change your environment?
Well, there are a lot of people, including Joe and others, who have open mind about this, and a lot of people are trying to find drugs against the microenvironment. For example, we now know that if you don't have immune system, your cancer will not progress. And there are a number of immune cells that are very involved, and wounding and inflammation are a very important risk factor. So we now know that if we treated blood vessel formation, wounding, inflammation, some aspect of immune system, cancer actually will not grow, or you can reverse it. So we are now working on therapies of both the cancer cell and the microenvironment.
So it'll come, to be continued.
It will come.
Okay, thanks.
All right.
The other thing that may be related to this too is tumor dormancy, where people have cancers, and you have cancer, it's treated, you're fine for 10 years, 15 years, and all of a sudden the cancer comes back again. You know, what were those cells doing for 15 years? They were there, and they were obviously being asleep, being held in this suspended state by their neighborhood, and then for some reason that we don't understand, broke out and shifted back. So this being able to revert back and forth is really probably very important in terms of what we see in women and people with all kinds of cancers. Our last speaker is going to, in some ways, is pulling these both together. You know, the question is, how do you get all these mutations? Some of them you inherit, but the majority of breast cancer is not inherited. Only 5 to 10% are people who inherited a corrupted gene, so to speak, from their mother or father. And the majority is, as Mina pointed out, things that we get exposed to. One of which, of course, is radiation. And that can both induce cancer, but then it also can treat cancer. And many of you know that we use radiation and lumpectomy and radiation to treat breast cancer. And so it can have effects on both sides of this paradigm, both screwing up the cells and then also may have effects on the environment the cells are in. And that's what we're going to hear from Mary Helen Barcellos-Hoff.
I feel kind of like the Monty Python skit, you know, now for something completely different. Except it's not going to be all that different. I'm going to talk about cancer as ionizing radiation, which we all recognize as a cancer-causing agent. Now, I want to assure you, first of all, that it's not a very good carcinogen. But unfortunately, as a human population, it's one we know when we've been exposed to. And we have a very large cohort of people who were exposed to ionizing radiation as a result of the atomic bomb detonations during World War II. And those people have been followed, and from that population where we've constructed radiation exposures, we can show that there is an increased risk of developing cancer at high doses. Now, what is a high dose and what is a low dose is another topic that we could go into quite extensively. But what we're going to talk about today is just so we can show that radiation causes cancer at certain doses. How does actually radiation cause cancer? What do we know about cancer from what Joe and Mina have talked about in order to inform us of what radiation is actually doing? So I'm going to talk about kind of this basic paradigm. Joe introduced the idea very clearly. Mutations in cancers are important to causing the cancer and can also be targets. What we think with radiation is that radiation causes this kind of DNA damage that leads to mutations, and therefore that ends up in cancers. So irradiated cells cause cancer. Right? That's those are cells over there that have been irradiated, and those little green spots are DNA damage localization. But what we heard from Mina is that tissues are essential partners in carcinogenesis, that the microenvironment is composed of all these other cells, including the basement membrane and these soluble factors that are required for carcinogenesis actually to progress. So it can be suppressed by normal tissues, as Mina demonstrated with her Rouse sarcoma virus experiment, where she injected the virus into an embryo, showed there's no tumors that formed. But it can be promoted by damaged or remodeling tissue. So you can come in and generate a tissue that supports carcinogenesis. So what we've studied in my laboratory is how we can take these two different ideas, the idea that you need a seed, i.e. that cell that has the mutation in it, and you also need the right soil. So this is an old hypothesis, seed and soil. Goes back about 100 years, and it's an essential part of how we think about cancer and how we think about radiation's action as a cancer. So these pioneering studies by Beatrice Mintz, Barry Pierce, Judith Folkman, and Mina Bissell have really emphasized how we now have to put these two pieces of the puzzle back together. So irradiated cells become cancers in the context of tissues. And so the next question is, really, how do irradiated tissues become cancers? So we're going to take that seed and we're going to put it into the right soil, and we're going to ask, how does radiation affect one versus the other? One of the ways to conceptualize this overall framework is that what we're doing is we're coming out of a period of reductionism where we studied the components, the molecular biology, all the individual genes and what they're doing, and we created a catalog. The human genome, which was initiated at Lawrence Berkeley National Lab, is one of those kind of projects. Joe mentioned the tumor genome project, another one, where we're cataloging the individual components. And then Mina talked about how these genes have to be placed into an appropriate environment, the cell biology, how cells talk to each other. And then, as Susan, I think, pointed out quite correctly, we have to think about the organism, the whole physiology of the individual. And in order to do that, we now have to link these different levels of organization. And we do that with what's now called systems biology. Now, really, this is an a new word for an old field, which is how you link all these different levels of organization. But one of the reasons that systems biology is so interesting is we begin to ask the question, how is the whole greater than the sum of the parts? And there is an idea that what distinguishes a complex system from a merely complicated one is that some behaviors now, kind of what Mina was talking about, how the cell behaves when it grows in that three-dimensional matrix, how those behaviors emerge as a result of altered relationships between the elements. And that's what makes the whole greater than the sum of the parts. We have the parts listing, but now we have to put it back into a context. So here's a very simple example of what we mean by emergent behaviors. A person breathes, a lung exchanges gases, but a lung cell, that individual part of the tissue, does neither. So you could consider breathing as an emergent phenomena, a property of the system, of the organism, rather than of the parts. So that's what we're going to be looking for when we now think about cancer. Because our hypothesis that we've been testing is that cancer risk is really from radiation, ionizing radiation, which we know causes DNA damage, which we know causes mutations, is actually a system rather than component driven. And I mean that it's not just a function of those mutations, but it's a result of some other, so the complex effects of radiation on the whole organism. So another way to put this is, does radiation induce emergent phenomena? That is, malignant behaviors that result from altered interactions and signaling. So how are we going to test that idea? Radiation is going to induce altered interactions. It's kind of like Susan's analogy. If you take one of you, stick them in an isolation booth, what do we really know about that individual cell? I'm going to say that actually what we know about individual cells is, like Mina was describing, a result of those interactions between cells. So we're going to use that same assay that Mina described in her talk, where you put cells, normal cells, into an extracellular matrix, and you're going to ask them to perform what they normally do. Their normal behavior in a mammary gland is to form this hollow sphere, which is called an acinus. But if you take tumor cells, as Mina showed you very nicely, and put them in the same system, then you have this disrupted morphogenesis, this altered interactions between cells, and that can actually be used as a way of measuring how radiation affects these cells. And we're going to do this, so always little lightning bolts in a radiobiologist's view is radiation exposure. So don't think electricity. Here we're talking about ionizing radiation. And I'm going to add this very interesting protein I study called TGF-beta, which does not stand for Thank God It's Friday. It's, although my sister thinks it does. It's a protein that mediates interactions between cells. And we find that in normal mammary gland, it's actually induced by ionizing radiation. And so here's our panel of very pretty pictures, which now show you if you take normal human mammary epithelial cells, expose them to radiation, and then add this growth factor called TGF-beta, that we get that kind of phenotype that Mina just described to you. So on the top panel, we're looking at sets of epithelial cells that are stained with different proteins that are all indicated down here. But what's important about these are all hollow spheres. They all have this nice protein localization at their surfaces and in some cases between the cells. And that's appropriate. These are the right places for these proteins to be. In contrast, when you expose the human mammary epithelial cells just once to radiation and then culture them in the presence of TGF-beta, you get a very different organization of these cells. They go from being hollow spheres with a single layer of cells to a larger group of cells, right? You can see there's more cells here. And they're completely disrupted. The way the proteins are expressed, they're either lost or they're expressed in the wrong place, completely differently. And so what this is that radiation that we expose that individual cell to, and then what we're looking at are all the daughters of that irradiated cell, has altered the relationship between cells, and that the irradiated cells have remembered that at least when they've been exposed to TGF-beta. So that suggests that radiation cancer might be an emergent phenomenon, right? We've got our cell assay that suggests that there's disrupted morphogenesis, which Mina demonstrated very nicely is associated with cancer, and it's indeed what pathologists look for in a tissue section. But can you now take this idea and transplant it into an animal model where you can actually see, I told you at the beginning, it takes a tissue to make a tumor. I've shown you cells in culture individual cells. And now the question is, can you perturb that interaction more sufficiently to actually cause cancer? So this is the classic mammary gland of a mouse. It's always an oval. And as Susan showed you in her slides, there's this little epithelial bud, right? Now, one of the ways we can take advantage of the mouse is that we can separate the epithelium from the stroma or the adipose tissue, all these cells that are out here that are just pale blue in this particular slide. And we do that by coming in at 3 weeks of age and just clipping out the epithelium before the hormones of puberty have induced growth. Now, if you then take and put in a aberrant tissue called, it's a gene that is involved in many, and in fact 50% of all cancers, called P53, we take a little fragment of that from another mouse, and that tissue now will grow out. So we've created a chimera. Here, the host is just wild type, and then the epithelium is P53 null. Now, we know this P53 is important in breast cancer, and what I find interesting as a model is that it has many of the features of human breast cancer. It develops aneuploidy, it develops a ductal carcinomas in situ, a DCIS-type lesion, has a very long tumor latency. One of the things we have to keep in mind is that women really, the majority of women, don't develop cancer until after 50 years of age. So we want a mouse model that does that. And it develops a diverse group of tumors, just like Joe indicated. Human cancer is diverse. You don't want to see the same cancer over and over again in your mouse. So what we did here is we first made this tissue chimera, and then we took this group of mice, and half of them we just left alone, and then another couple of groups we irradiated with very tiny doses of radiation, 10, 50, and 100 centigrade, and again we can talk about doses later. And then we transplanted them after we irradiated them with unirradiated P53 null tissue. And the question was this: radiation is a carcinogen. Radiation can cause cancer by inducing mutations, but can it change the interactions between cells? So, in this case, we didn't irradiate the P53 null tissue. It has its own machine for making mutations. So, in this case, radiation is not causing mutations in the target cells, the epithelium. It's only changing the host, all those other cells.
The neighborhood.
The neighborhood, exactly. Right. That's like, you went from South LA to Hollywood, or vice versa. So we're trying to modify the neighborhood. And when we did that, we have found a very striking result. We found that, okay, these animals will develop cancer. At about 70% of them will develop cancer by a year of age. But when we had pre-irradiated the host with this dose of radiation, regardless of the dose, we got 100% of the animals developing tumors. So that suggests that radiation can indeed act through all these other cells, through the microenvironment, to alter it, to make it a bad neighborhood, run it down a little bit. Now that in itself is very interesting, but as Susan pointed out, what good is that to preventing cancer? What if you had a population of people who were irradiated? How could you manipulate this environment? Now, I showed you on my other slide that TGF-beta, this protein, this magical protein, was important in driving malignant behaviors in the cell culture dish. So what we did here is we took the same assay, we transplanted them into a mouse that had less TGF-beta, because I said, okay, TGF-beta is acting as a bad actor here. It's causing cells to undergo disrupted morphogenesis. What if I take it out of my irradiated host and see what happens? And what we found is a dramatic reduction in the ability of radiation to promote cancer. So that says that radiation is acting through the host to promote cancer by inducing, at least in this particular model, this one protein, TGF-beta. And one of the things we already know is that TGF-beta polymorphisms in human populations are associated with increased cancer risk.
Polymorphisms means screwed up TGF-beta.
Well,
genes.
screwed up
genes. The
there
genes
are
that
varieties. Yeah, variations
right.
on the theme. I'm sorry.
Right.
Thank you for it. Okay, so radiation can actually cause an increase in the risk of developing cancer by altering simply the interactions between cells. So what does that tell us? Why is that important? Well, it says a couple of things. One, it says that radiation both affects the DNA and the genes and the microenvironment. Okay? So that's a new idea in Well, it's actually not a new idea. I found out that somebody had done this same experiment in the 1950s with thymuses. But that's typical of science, is that ideas come and go, come and go, come and go, come and go. So in this case, it's come and gone, and now we have further evidence that this effect through the microenvironment is important. Now, what that also tells you is that the effects in the microenvironment are avenues for protection after the radiation exposure. You can't do anything once you've damaged your DNA and you've got a mutation. I don't know what your mutations are versus my mutations versus the guy over there's mutations. But I can come in and say, well, these signals from the irradiated tissue are important in the development of cancer. And maybe, just like Joe said, we could target those signals. We can target these signals in the irradiated host. And I think that's an important thing to consider. Finally, I think that one of the things that this implies is that predicting how radiation affects health will require a better understanding of this two-compartment, this dialogue between cells, the dialogue between the individual and its environment. And that in doing so, we're going to have some new perspective on how radiation acts as a carcinogen and when we should be worried about its action as a carcinogen versus its action as a therapeutic. So with that, I'm going to acknowledge here not only the people who've done the work, which are many and myriad, but also the Department of Energy. We're also supported by NASA, and we're supported by the NIEHS Breast Cancer and the Environment Centers. So thank you. I have a question. And the question from Susan is?
I have one question, and then I'm going to bring everybody up so that you all can ask questions. So if this is how it causes cancer, but we also use it to cure cancer.
Yep.
Do you have any idea how that works?
Well,
what's the difference?
Well, dose is very important. It's the same thing as if you sit out in the sun for 5 minutes versus 14 hours. You're going to feel an effective dose, right? And then the second idea is that dose rate is important. So chronic irradiation, like if you sit out in the winter sun for 14 hours, maybe it's not so bad as sitting out during July, unless, of course, you're in Berkeley, in which case there is no sun in July. So I guess the thing that so one of the things that we found is this radiation-induced TGF-beta also occurs in cancer treatments and is actually acting to suppress the good things that radiation does, which is to kill cells. So we now have evidence that we've suppressed TGF-beta in our cancer model, but we can also suppress TGF-beta in tumors, and it increases their response to therapy. So we've learned something useful.
Good. Good.
Okay.
Well, at this point, let's put up the lights. Let's bring our panel up and give you all a chance to ask questions.
Wake up.
That's why we're turning on the lights, Mina. So we're going to have you ask the questions. We found that doing microphones here is difficult. I will repeat the question, and then we can answer it. We'll get any or all of them to answer it. I can't imagine that nobody has any questions. Yeah.
So
what created the Marin Cancer Cluster? I can actually answer a little bit of that. Breast cancer tracks very well with socioeconomic level. The higher your socioeconomic level, the more breast cancer, regardless of ethnic group, within every ethnic group, and even with countries. So breast cancer is more common in Europe and the United States and less common in Africa and South America. And we know some of the elements and not all of the elements. I mean, one of the things is probably age of first childbirth reduces your risk. Things like hormone replacement therapy, which you're much more likely to take if you're, a higher socioeconomic level. But apart from that, we don't fully know that. But all of the hot spots, Long Island, Orange County, Marin, are also high socioeconomic level. So there may be environmental factors as well, but that's a huge part of it. You want to say something?
I like to add, somebody asked me this question actually yesterday. In addition to what Susan said, one of the other things is that pregnancy is one of the most wonderful risk factors, either good or bad. So when you have children early, pregnancy is protective. When you have children late, pregnancy, in fact, is not very good for you. It goes the other way. And many of these women in these higher social economic things have children very late. And we know that alcohol, in fact, exacerbates the rate of many kinds of cancer, prostate as well as breast. But if you take folic acid and you take 2 glasses of wine, you're just fine. But if you don't take folic acid and you have 4 glasses of wine, you're in trouble. So as she said, you know, these people probably have wonderful lives and do whatever. And, you know, it's a very complex question. The other thing is, I don't know what these clusters mean. You know, statistics is
well,
not...
they have done. They did a very careful study they did a study in Marin, they did a study in Long Island, and they really were not able to pick up any one thing in some of these clusters. So, you know, we'd like to think, we really want to find one thing in the environment we can blame it all on, but so far we've not been able to do that.
So can I add to just because the last grant that I acknowledged from the NIEHS, Breast Cancer in the Environment, actually was stimulated by the advocacy groups in the Marin community who brought this question, exactly the same question, to the NIEHS and said, what is it about our environment? As a result, the NIEHS has funded 4 centers across the country who have a unique structure, to say the least. On the one hand, there are biologists like myself who are studying the biological determinants of maturation of and puberty in rodents, in mice and rats. On the other hand, we are linked with epidemiologists who are studying the maturation of young girls, actually the transition through puberty, and identifying what actually dictates that transition. With ionizing radiation, there's good evidence, in fact, it's probably the only evidence, that